no matter how far. With that, I request Saurabh to introduce the speaker.
Is it not working? It's working. Can you hear me? Okay. So it's my great pleasure to introduce Professor Mriganka Sur, who is the speaker for our 17th Ramamurthi Memorial Lecture. Professor Mriganka Sur is very like an integral family of NBRC. Since I joined as a faculty at NBRC back in 2012, I have seen him almost every year since then, except the COVID period of time. So Professor Sur is a Newton Professor of Neuroscience at MIT, and he's a founder director of Simon's Center for Social Brain at MIT. And he was, prior to this appointment, he was chair of MIT Department of Brain and Cognitive Science for 15 long years. And he has made seminal contribution in our understanding of the visual plasticity, and particularly working on how the cerebral cortex and cerebral cortex circuitry regulates the visual plasticity. And some of these experiments we have discussed in our developmental neurobiology class about the Anna Rowe and Mriganka's experiment on visual plasticity. So he has received numerous awards, and to name a few, like he received NIH Brain Initiative Inaugural Award in 2014, and he's the elected fellow of the Royal Society of UK and the National Academy of Medicine of the US and the American Academy of Arts and Sciences and the American Association for Advancement of Science. And with this short introduction, I would like to invite Professor Mriganka Sur to deliver the 17th Ramamurthi Memorial Lecture. Please join me to welcome Professor Sur. Asking questions.
Okay. Is this on?
Yeah.
Okay. Yes. All right. Okay. And okay. Very good. Thank you very much, Saurabh. And thank you very much, Professor Sengupta, and everybody here at the National Brain Research Center. Thank you for showing up. It's my great pleasure to give today's lecture and to also spend the day with you to see how NBRC has evolved and matured and continues to progress. And I regard myself as a strong well-wisher of your center. I'm going to talk today about some work that my lab has done over the last many years that has led to a major success, actually, just a year and a half ago, which is the approval by the Federal Drug Administration in the United States of the first treatment, pharmacological treatment for a devastating brain disorder called Rett syndrome. So I'll build up to that as I go along. And then we'll talk about what else, what next. So my laboratory works on the cerebral cortex of the brain, which I think most of you know something about. You know where it is, and some of us use it. Sorry, that was a joke. And but the cerebral cortex is a complex entity that has about 200 different regions. And for development, an amazing issue is how does this complexity of the cerebral cortex come about? And so in one slide, here is a very brief primer going back many years. Now you have Dhananjay here who can tell you in great detail, much more sophisticated detail, how the cortex actually develops. But here's a simple view. Very early in utero, even in the first month after the embryo begins to be formed, there are centers that lead to gradients of transcription factors that interact with each other to lay down the primordial substrates for different brain regions, which then begin to attract axons or wires from other brain regions. And once these axons have connected, then synapses or connections between neurons begin to form. And then in the postnatal brain, in the human cortex, these synapses continue to form, but importantly, they are remodeled to create networks and cell assemblies that persist into adulthood as substrates for learning and memory. That's a big one-sentence summary of decades of work that has been done and is yet to come. Importantly, cortical areas and pathways form early, whereas synapses and circuits consolidate later and are shaped by mechanisms of plasticity. And what is plasticity? Plasticity is the adaptive response of brain cells and circuits to changes in inputs. Okay, that's a simple definition, but we'll build on that. And I'll start with the proposition that many brain disorders, particularly disorders of development, that have their origin in genes that make the brain, you also need genes to make plasticity possible. And many disorders of brain development arise from deficits in synaptic development and plasticity. Hold that thought, okay? So today I'm going to tell you about three things. And in a way, it's kind of, as I reflected on what should I talk about, I'll tell you how my own work has progressed over the decades, starting with some of our early work on plasticity. I've been in plasticity for my entire career now, spanning 4 decades. How that has led us to think from reduced systems to more sophisticated systems of plasticity, how it works, and how we have used this understanding to understand mechanisms and come up with therapeutics for Rett syndrome. And I'll tell you about Rett syndrome, okay? So we'll start from about 40 years ago and we'll end in the present, more or less. All right. So first, mechanisms of plasticity and the two forces of Hebbian plasticity and locally coordinated plasticity mechanisms, how they shape changes in brain cells and circuits. So many years ago, almost 40 years ago, when I started my lab, I started with a very simple question, which is, what is visual, let's say, about the visual cortex of the brain, which sits at the back of the brain, which gets input from the eyes through the visual thalamus to the visual cortex. What we see starts with the eyes, but is really created in the brain. The eyes just give you a set of pixels, but from that, the brain creates vision by organizing the subsets of vision via elementary properties such as orientation selectivity and edge detection, and then more and more complex objects like faces and places and things, okay? So to answer this question, how does the visual cortex become visual? We rewired the brain. I made visual inputs that normally go to the visual thalamus and to the visual cortex. We made them go to the auditory thalamus, which processes hearing, by removing the inputs that normally go from the ears to the inferior colliculus of the midbrain, to the medial geniculate nucleus of the thalamus, to the auditory cortex. And we did these experiments in newborn ferrets. This is a picture from about 540 years ago, made by Leonardo da Vinci. It's called Portrait of a Lady with Ferret, and it hangs in this museum in Poland. And I actually went to this museum to take my own picture of this very famous painting. And you can see at a glance why a ferret is a wonderful animal model for vision. It has frontally placed eyes, and importantly, a ferret is born very early in development, at the stage of about the first trimester of gestation in humans. So you can do things to a postnatal ferret that you have to go in utero for the vast majority of animals. And if you want to rewire brains using very humble tools, not genetic tools, but physical surgery tool, that's a big advance. So we took newborn ferrets, which are about as big as half my finger, and the brain is about as big as my nail. And in these ferrets, we removed the inputs that go from the auditory thalamus from the auditory midbrain to the thalamus. And we let these animals grow up. And we discovered that the retina now projects to the auditory thalamus, thereby providing a pathway for vision to activate the auditory cortex. Okay? Much later, we figured out that that's because this auditory thalamus now expresses genes, ZIP1 and ZIP4, that are normally expressed in the visual thalamus, but it allows fibers from the eye to recognize this novel structure and project there. We let these animals grow up. Remember, now they have this novel pathway, and it enables us to answer our question. What is visual about the visual cortex? Would now vision driving the auditory cortex make the same cells that normally exist in the visual cortex? And the answer is yes. So we did many experiments in these animals, rewired at birth, grown up to adulthood, and while the normal visual cortex has these orientation selective cells and maps of orientation selectivity, so does the rewired auditory cortex. And I remind you that the only thing that the cortex knows about the outside world is contained in the electrical activity of these wires that go from the thalamus to the cortex. Okay, that is the only knowledge that the cortex has during development of what is driving me. And in the rewired animals, it is vision and the properties of vision in space and time that are driving these wires that project from the auditory thalamus to the cortex. And this activity makes orientation selective cells, and it changes the circuitry of the auditory cortex. Normally, in visual cortex, horizontal connections link neurons with the same orientation preference. In the auditory cortex, horizontal connections link neurons with the same sound frequency preference. In the rewired auditory cortex, the neurons connect, the connections link neurons with the same orientation preference, similar to the visual cortex, but very different from the auditory cortex. And this change in circuitry is made possible by the electrical activity of these inputs. There is no other information, which leads to perhaps the most important question in our field. How does electrical activity shape synapses and circuits? This is the principal way by which the world in which we live in shapes our brains. And I just showed you an example of how the nature of the world shaped the circuits that process that world, albeit in a rewired brain. It's a model. All biology, all life sciences relies on manipulations and models to probe mechanism. Okay? So now, here's an important idea attributed to Don Hebb from a paper that he wrote more than 70 years ago. And Hebb was a psychologist, and he said, here's one way by which activity can shape circuits. Neurons that wire together, fire together. He actually wrote a lot more than this, but this is my interpretation. But there's a problem with this, which is that if you have, let's say, one neuron that continuously fires and is followed by this neuron firing, so that's two neurons are firing one after the other, you wire them together. The stronger this connection, the more likely that adjacent neurons will end up randomly being paired with this postsynaptic cell. So it's been well known that unregulated Hebbian plasticity leads to saturated excitatory synapses and unstable assemblies. Ultimately, all these inputs will be wired up and they will all be firing all together. So this cannot be the way that the brain is wired up. So you need ways to change and organize these connections. So one of these ideas is synaptic renormalization, which is that you have to down-regulate certain connections which have been strengthened And synaptic scaling is a prominent form of renormalization. In fact, the dominant idea of why we sleep. You know, we spend more than a third of our life sleeping. Many animals, every animal sleeps, including fish and fly and worms. Animals sleep even when they can be eaten when they sleep. So at great risk, sleep is built into our biology. Why? And it's thought that it's because we want to rest or this or that. I don't believe so. I believe the reasons are deep in the brain. One thing that sleep does is that it carries out synaptic scaling. If during the day you have two synapses that have been strengthened over time in this ratio of receptors, and of course, the way that synapses strengthen is that you first activate NMDA receptors, which are the detectors of pre and postsynaptic Hebbian driving, and then you insert AMPA receptors. And I'm going to assume you are neuroscience students, so you know at least a little bit of what I'm talking about. Okay? I asked Saurabh, who's going to be my audience? And Saurabh said, oh, they're all neuroscientists. So I said, wonderful. Then I can talk about science. Otherwise, I was worried that I have to talk about, you know, something at like science and humanity or something. And I'm no good at that. I'm just going to tell you about science. So we are going to now take a deep dive. Okay, so now, when these receptors, when these synapses strengthen because they have got an AMPA receptors in them, these receptors have been going up during the day, and then synaptic scaling means that you change, you remove receptors, but you keep the proportion similar. So this proportion is 2:1. And that's what synaptic scaling is. And there is a lot of evidence this is what sleep does. But there is another form of normalization that may play together with Hebbian mechanisms to create circuits. And I call that locally coordinated synaptic plasticity. It's another form of renormalization which leads to strengthening Hebbian inputs while weakening non-Hebbian synapses. And the idea is that when one synapse increases its strength, strengths of nearby non-Hebbian synapses should decrease. This has been an emerging idea in the field, and a few years ago we demonstrated that is actually a powerful way by which circuits can be created. So can we demonstrate this phenomena? I'm going to tell you about that in the next few slides. So here's a very simple experiment. It's simple in concept, and I hope I can explain it quickly. How might we study locally coordinated plasticity in awake mouse visual cortex? Almost all of our work is in awake mouse brain, and we use high resolution imaging to image single cell circuits and even single synapses and even single molecules inside synapses in vivo. Here's the idea. Suppose we have a single neuron in the brain, in the visual cortex. It has 4 synapses. Suppose we find a way to strengthen 1 or 2 of them selectively, and we measure it. What happens to the others? And if there is locally coordinated plasticity, the ones we target should go up in strength and in size, and the ones we don't target should come down. Because then there is locally coordinated inflow of current and a conservation of the total input, but selective shaping of targeted inputs by strengthening them and down regulating inputs that are not targeted. Okay, that's the basic idea. We do this as follows. Here's a neuron in visual cortex, and it has 4 synapses. This neuron has a receptive field. It's a window on the world that is this dotted line. Okay, these 4 synapses, S1 to S4, have their receptive fields that are shown in the hazy orange and blue shade. And this neuron integrates all these inputs and makes a Gaussian so that its own receptive field is this dotted line. Okay, now we change the receptive field of this neuron by a Hebbian process. We want to change it from this dotted line to this, where this X is. So we shine a spot of light on that X, which leads to a EPSC in the cell. And when that EPSC happens, we make this cell fire by optogenetic means. So we have paired a input with a output to make a Hebbian synchronization. And the hypothesis is that it would strengthen those synapses whose receptor fields overlie that X, which are these two synapses, S1 and S2. And the synapses that are not stimulated would now weaken, so that these synapses would get larger and stronger, and these synapses would get weaker if locally coordinated plasticity is correct. And this is what we did. So we fill a cell, a single cell, we stick an electrode in it, and we fill it with channel rhodopsin. When I say we, I mean two very talented postdocs who now have their own labs, with channel rhodopsin, as well as with a calcium sensor that allows us to image the functional properties of this cell and all of its synapses, and a structural marker called mRuby. Okay, so here is the cell's receptive field, and then here is where we want to move the cell's receptive field to. So every time that we can, for 100 trials, we shine a spot of light here, and we stimulate this cell with blue light. Okay, so that's the pairing. It's called the spike time dependent plasticity. And after we do this for a while, we can show that the neuron's receptive field has actually moved here. And what is the underlying mechanism at the level of single synapses? And we can measure these synapses. I remind you that the cell is about 10 to 20 microns, which is about 1/10 to 1/20th the width of a human hair. And a single synapse is about 1 to 2 microns. It's about 1/50th the width of a human hair. But we can measure them in vivo in an awake behaving mouse. Okay. Okay. So here is a single dendrite of that same neuron that we filled. Okay, I'm going to compress a lot of work into just a few slides. So the spines that are potentiated, so structural LTP, and they are shown in red. But there are other spines that are actually depressed that are shown in green. These are the spines that we strengthened. We didn't do anything to the green spines. They just happened to shrink by themselves. And every red spine is surrounded or is in close proximity to a green spine. And we can ask, where are the receptive fields of the red spines? And remember, this is about 1 to 2 micron, and we are scanning with our laser. This is a mouse whose head is held and is under our 2-photon microscope. But if we keep everything very tight, and we are mapping the receptive field using a mechanism called reverse correlation. I can tell you about it later. So in this way, we can plot the receptive field of a single spine, a single synapse, very precisely. The red spines all have their receptive field on top of the X, and the green spines have their receptive field around the X, arguing that the spines that have gotten bigger, fatter, stronger have been induced to do so by our mechanism. And the spines that have gotten weaker have become so by virtue of the cell coordinating its plasticity. So that's the basic idea. Plasticity involves two phenomena. You induce a change, and the cell responds to that change. You cannot have one without the other. And why does the cell respond? Because it wants to normalize its inputs. Some people call it homeostasis. I think Saurabh will call it homeostasis. I don't like the word. Homeostasis means you go back to some original state. There is no original state. We have changed the cell's receptive field, but we have conserved the total amount of current flowing into the dendrite. Okay, so therefore, very quickly, surrounding every enlarged, strengthened spine within 50 microns are spines that have gotten weaker, So synaptic plasticity is locally coordinated on dendrites, and the issue is, what is the mechanism? And I'll make a long story very short and say one of the critical molecules that we think that is involved in plasticity is a molecule made by a gene called ARC. It's an immediate early gene. It's upregulated in excitatory neurons by synaptic activity. We did not discover this. It's been known for 30 years. And it was discovered in the hippocampus, important for learning and memory, by electrically stimulating the hippocampus. Electrical stimulation, it upregulates ARC. High frequency stimulation, it upregulates ARC. Long-term potentiation, it upregulates ARC. And it took another 10 years to figure out what does ARC do. And ARC does something amazing. ARC removes AMPA receptors from synapses. One of the most important mechanisms that is a marker of activity is actually a mechanism for reducing the impact of activity. The brain has gone to amazing lengths to make sure it doesn't go into hyperdrive. Okay, it's an amazing thing. So ARC down-regulates synaptic efficacy, yet it is the most important, one of the most important markers in the brain, along with CFOS and JUN and other immediate activity genes of marking neurons and loci of activity. What does ARC have to do with our model? Everything, we think. So we think that when a synapse is strengthened, ARC leaves this synapse and migrates to adjacent synapses, and there removes AMPA receptors to induce them to be depressed. And so that ARC translocates from potentiated spine into less active spine and regulates LTD by removing AMPA receptors, and it acts as an inverse synaptic tag. And this spine gets strengthened because of CAM kinase and many other mechanisms that have been involved in the study of learning and memory. So then we expressed in single neurons in vivo in awake mice markers for ARC and markers for GLUA1, which is AMPA receptors. And again, to make a long story short, potentiated spines have less ARC and more AMPA receptors, shown here. Larger spines have more AMPA receptors, and larger spines have less ARC. Depressed spines have more ARC and less AMPA receptors. And finally, if you knock out ARC knockdown increases GLUA1 enrichment, but it prevents the plasticity, the shift in receptive field, and it prevents the spatial organization of fattened and thin spines. Okay, so very quickly, cortical circuits are shaped by electrical activity during development using mechanisms of Hebbian plasticity and renormalization. Locally coordinated plasticity in dendrites involves ARC expression in depressed spines and ARC-mediated AMPA regulation. Okay? What does this have to do with circuit-level phenomena and eventually brain disorders? About 40 years ago, Hubel and Wiesel won a Nobel Prize for showing plasticity in the intact brain. Okay, they also showed orientation selective cells. They won it for two reasons. And this plasticity in the visual system is induced by closing one eye during a critical period of development. Mice or monkeys or cats and human beings need vision during an early period. If a child has amblyopia, meaning that they have either exotropia or esotropia, the two eyes are not aligned. And if you don't fix it within the first 2 years of life for a human being, the two eyes will never align. If you fix it later, and the child will never have binocular vision. You see in depth because each object in space is seen in slightly offset by one eye or the other. Okay? And that requires the two eyes to be matched. If you close one eye during a critical period, for a mouse, it's only 4 weeks. Then if you record neurons in the visual cortex, they will all be driven by the open eye. They will not be driven by the closed eye. Even one day, even a few hours of closure does this. So this is now the opposite of what I showed you. I showed you how to increase the strength of synapses and other synapses decrease. Now we are decreasing the drive to neurons, which leads to reduction of drive after 4 days of closure. But then if you keep the eye shut, you find something amazing, that so if you keep the eye shut, then the closed eye drives neurons less and less. But then if you keep this eye shut, you find that the open eye now drives these neurons more. Nothing has been done to the open eye. It was open, it remains open. Yet, by closing the other eye, you increase the drive from the open eye. And it's an analog of how you induce a change, reduce inputs, and the brain responds to it by reducing the drive, but then increasing another drive in order to compensate for this reduction. This is the deep principle of plasticity, okay? And you can derive what's called the ocular dominance index, which is the neurons drive from contra I minus ipsi I divided by contra plus ipsi, and the net result of closing one eye for 4 days and 7 days is a progressive reduction in this ocular dominance index, which I'll come back to in a minute. Okay, so what does this have to do with ARC? Everything. If you remove ARC from a mouse, then this normal reduction of drive after 3 days of closure does not happen. And it does not happen because there is no removal of AMPA receptors as happens in a normal mouse. Okay? But if you close the eye for 7 days, then this normal increase of responses from the open eye also does not happen. Okay? So it prevents the subsequent scaling up. And so ARC is crucial for visual cortex plasticity on for bridging, feed forward, and feedback plasticity. Neither of these happens when you remove ARC. In the next 20 minutes, I'm going to now take you to what does this mean for brain disorders, okay? So brain disorders such as autism or neurodevelopmental disorders, they have been increasing in diagnosis, but they have strong genetic origin. They have strong genetic risk factors. Some of these risk factors are single genes, but the vast majority of the risk factors are complex multigenic. So rare and common variants both contribute to autism risk. As a rule of thumb, rare variants, a few genes or a single gene, have large effects, whereas common variants, where there are hundreds of genes that lead to a phenotype, each gene has a very small effect. So these single gene variants provide a powerful window into making animal models, because you can knock out one gene, and into mechanisms. What does this gene do, and how does it change brain function so as to create the developmental disorder or autism? And there's a major gene discovery effort underway. The Simons Foundation supports a lot of this work. I direct a center funded by the Simons Foundation. At last count, from various cohorts, the simplex collection is where only one person in a family has been diagnosed with a developmental disorder or autism. 3,000 such people were collected. By definition, it will discover de novo variants, meaning new variants. It is not familiarly transmitted. One person in the family. In contrast, triplex, 3 people in a family, mom, dad, and son, or dad, son, sister, all are diagnosed with autism. That leads to the SPARC collection, and that leads to genome-wide association studies for looking at many, many genes or even variants that can contribute to autism. Okay, so these are various ways to look at this. For the last 15 years, we have studied one disorder called Rett syndrome with these principles of plasticity in mind, and I'll tell you about it in a minute. But first, if there are 1,400 genes at least, and at least 500 or 600 of them have high confidence, if you go to this database, Safari gene, 600 genes today have been linked with neurodevelopmental disorders, including autism. How can it be? What might these genes do? So, can we form unifying hypotheses about the function of autism genes? Gene discovery is just the first step. The critical next step is to find out, what do these genes do? Third, how can we use that information to treat the disorder? What do these genes do? And here are some facts. A large number of autism genes, autism spectrum disorder genes, are expressed at synapses. Here are some of them from a review article by Mike Greenberg. Defects in activity-induced gene expression contribute disproportionately to autism. About 60% of the autism transcriptome is activity dependent, as opposed to 3% of all genes that are expressed in the brain. So electrical activity or neuronal activity is a very important driver of the expression of genes that contribute to autism spectrum disorder, which leads to the idea that components of activity-dependent plasticity, namely what I've just told you, Hebbian plasticity and renormalization, are very likely affected in autism spectrum disorder. Okay? So feedback plasticity in visual cortex neurons is an expression of locally coordinated plasticity. Plasticity mechanisms engage a number of neurodevelopmental disorder autism genes. I haven't told you anything yet, but now I'm going to use this fact to discover this fact, and as it relates to Rett syndrome. Okay? So now I'll tell you about Rett syndrome in the last 15 minutes. Rett syndrome is a devastating disorder. I know several families with Rett syndrome. It's very rare. It's rare, it's 1 in 10,000 births. It's caused by de novo mutations in the MECP2 gene, which lives on the X chromosome. MECP2 mutations are also linked to other neuropsychiatric disorders. And because it lives on the X chromosome, boys have one X and one Y. And if they have a mutant X allele, boys don't live. Within the first year, they are dead, or they have a very severe condition called congenital encephalopathy. Girls have two X chromosomes, they have a mutant allele and a normal allele. Girls are mosaic for MECP2. Some cells have the mutant protein and other cells have the normal protein because of random X chromosome inactivation. Okay? So 50% of the brain has mutant MECP2. What does that do? By 1 or 2 years of age, they lose their speech. They begin to have this typical hand movement. They have respiratory distress. They have cognitive deficits. They have anxiety. They are susceptible to seizures. They have smaller heads. Their brain is smaller. Their neurons are smaller. And that was the first clue that we could think about this disorder in a different way than earlier. I'll play a little video for you. Many people put up their videos online, and I hope you can hear it. एक minute.
Left her unable to talk or communicate, but life hasn't always been like this. In 2007, Gabby came to the world a healthy, happy baby girl. The first year of life with her parents and two siblings was for the most part on track. But around a year and a half, her mom noticed a serious progression in her name. She parents took over to say like God and mom and pop and all that, dad, she would say, yeah, and then all of a sudden she stopped talking. The constant hearing of this led doctors and children to test families specifically as syndrome. It is a neurological disorder occurring mostly in girls. Rett syndrome is caused by mutations on the X chromosome.
I think that's okay. Okay. Very quickly. The MECP2 gene has different regions. There is a methyl binding domain and a transcription repressor domain. It's a single gene disorder. In the New England area where I live in Boston. So in the northern states, northeastern states, there are about 250 girls who have Rett syndrome. And I know because we ran a clinical trial involving these girls. There are multiple mutation types. There can be mutations in all of these regions, but there are 8 hotspots. There is functional abnormalities in multiple brain systems. MECP2 binds to methylated DNA across the genome, and it influences many genes, but it also regulates microRNAs and interacts with RNA polymerase II. And loss of MECP2 affects multiple developmental stages. The gene was discovered by Zogby in 1999 using just linkage analysis. But soon after the gene was discovered, knockout mice were made about 25 years ago. They were made by different labs, including my collaborator, Rudolf Jenisch. MECP2 expression, we know from human studies and mouse studies, starts early, but then plateaus and remains high throughout life. And the mice recapitulate many of the human phenotypes, including if you hang them by their tail, they will clasp their hind limb. And they have deficits in anxiety and seizures, etc. We know a lot about MECP2 mechanisms. It represses or de-represses transcription by binding to 5-methylcytosine or 5-hydroxymethylcytosine, which are complements of each other. It acts post-transcriptionally via regulation of microRNAs. But in 2007, a paper appeared from Adrian Bird's lab that made me switch to work on Rett syndrome. And this is what Adrian Bird's lab did. They made a mouse in which they held the gene in check. So by 4 weeks, 5 weeks, the mice began to fall sick. By 5, 6 weeks, the mice are very sick because they don't live for more than 10, 12 weeks. But then they turned the gene back on through the TET system, and the mice recovered. With whatever mechanism they could see, the mice lived longer, their breathing got better, they began to move better, the mice recovered. And this was a transformative event for the field because it said, this is a potentially treatable disorder. This is not a disorder of degeneration. This is a disorder of frozen development. This is a disorder of frozen development. It doesn't mean all developmental disorders are, but Rett syndrome is. I switched a third to half of my lab's effort to work on Rett syndrome because we hypothesized that synaptic maturation and maintenance are key aspects of MECP2 function, and synapses and circuits remain immature, frozen development in Rett MECP2 knockout mice, but can be induced to mature. Okay? I'll just compress a lot of work into the last part of my talk. What does MECP2 do? MECP2 binds to methylated DNA, but it also bridges to HDACs. MECP2 represses transcription. So then, when it's mutated, gene expression goes up, and certain genes, definitely the expression goes up. Importantly, binding proteins for IGF-1, which sequester IGF, and ARC expression goes up. But then, we and others discovered MECP2 also regulates production of microRNAs. So MECP2 inhibits the production of microRNAs, and when MECP2 is mutated, there are more microRNAs, certain microRNAs, microRNA 199, 214 are produced, which leads to reduction of gene products, and certainly products like IGF-1 or BDNF, growth factors, are reduced. Hence, the brain is smaller, the body is smaller. And very recently, from work from my colleague Rudolf Jenisch, MECP2 interacts with RNA polymerase II It acts as a positive cofactor for RNA polymerase II gene expression at genes that harbor CPG islands. And this too leads to reduced gene expression. So you can see at a glance, it has 2,000 targets. Some go up, some go down, and there are multiple mechanisms. How can we think about this? We can think about this at the systems level. And so MECP2 regulates molecules of activated dependent plasticity at synapses. Here are some of them. MECP2 mutations affect transcription of growth factors, like I just showed you, and MECP2 affects binding proteins. And so here is coming back to visual cortex plasticity. What are the molecules that regulate visual cortex plasticity now? And we did a screen many years ago. And to make a long story short, there are many molecules that are involved that change when you close an eye in a mouse during development, okay? But you can link them up. You can link them up through gene set analyses. And one of the most powerful pathways is an IGF-1 pathway, and about 30 genes in this pathway, including IGF-1 and binding proteins and the downstream signaling molecules such as PI3K and AKT and mTOR and S6. So during the critical period for visual cortex plasticity, IGF-1 levels are low and its signaling is low, and it enables visual cortex plasticity. If you increase IGF-1, and that is what happens to close off the critical period, the level of IGF-1 goes up, that abolishes plasticity. And coming back to MECP2 knockout mice, we find that IGF-1 is low throughout life. And here are some measurements of IGF-1 in MECP2 knockout mice of the message and of the protein. So here's our hypothesis. Our hypothesis is that decreased IGF-1 signaling in Rett syndrome leads to unregulated plasticity, extended plasticity, and immature frozen plasticity, which can be restored by exogenous application of IGF-1 or active IGF-1 peptide. So MECP2 negatively influences binding proteins, it positively influences growth factors through microRNAs that Anindya knows more about, or we were inspired by some of his work as well, and work in the C. elegans field to regulate a bunch of signals that are down-regulated in mutated MECP2 mice. But then you can restore some of these by exogenously adding IGF-1 or a peptide. It's a simple idea. And just to give you a little bit more about mechanism, IGF binding proteins are regulated by astrocytes. So in mouse, MECP2 knockout mice, there is high IGF binding proteins in astrocytes. Other labs have shown that too. And when you add a IGF-1, or importantly, a peptide of IGF-1, which is IGF-1 is a 71 amino acid protein. The first 3 amino acids are glycine, proline, and glutamate. It binds to the IGF binding protein, thereby freeing up IGF-1 to be acting on IGF receptors and its downstream signals. So that's the idea. And also, a bunch of other proteins are regulated by the IGF peptide. Very quickly, I'll give you a glimpse of how IGF peptide works. So here is our visual cortex plasticity model. And if you close one eye for 3 days in a 4-week-old mouse, the responses shift to the open eye. This is what visual cortex plasticity is that I showed you. If you do this in an 8-week-old mouse, the critical period is gone. IGF-1 levels have gone back up, and there is no effect of closing the eye. You close the eye for 3 days, then you open it back up, you do your imaging, you do your electrophysiology, there is no reduction of drive from the closed eye. You take an 8-week-old MECP2 heterozygous or homozygous mouse, and there is still plasticity. There is extended plasticity into adulthood. And then, if you treat these mice for 4 weeks, 6 weeks with IGF-1 or the peptide, you prevent the plasticity. You have made the cortex mature because the closing of an eye no longer switches responses to the open eye. Okay, I hope you got a little bit of that as to how we can use the visual cortex as a model within the MECP2 knockout mouse, which is a model for human Rett syndrome. But the logic of our thinking is that there is this powerful underlying mechanism of IGF, or the peptide, which acts through the binding protein to activate IGF signaling. Okay, and IGF upregulates AKT, it upregulates ERK, and it upregulates PSD95, which anchors AMPA receptors and increases synaptic transmission. So we have done many experiments, published in many papers, in which we have shown that while synapses normally mature from birth to about 8 weeks, that maturation is prevented in adult MECP2 knockout mice, but IGF-1 increases that maturation. And there are many, many measurements of synapses, of circuits, of systemic measurements of heart rate, respiration, etc. So all of this leads to the idea that the human disorder with this gene can be used to make animal models, and by application of basic neurobiology, we can come up with a hypothesis that can be used to develop a therapeutic which can go into clinical trials. And we published our paper in 2009, and there's a company called Neuron that actually had a version of IGF peptide that they had made for some other reason, actually for treating people who have stroke in order to prevent the brain damage after stroke. That didn't work. It's the same molecule with a methyl group added to prevent the metabolism of the molecule quickly. Peptides are notoriously metabolized within minutes. So this company had packaged IGF peptide in an oral emulsion to make it release slowly. And by 2019, they had published a double-blind randomized placebo-controlled study of trofinetide, which is really IGF peptide, in pediatric Rett syndrome. And these girls showed significant improvement on measures of Rett syndrome behavioral questionnaire, and the clinical global impression scale. And then there was a major phase 3 trial done by a company called Acadia, which acquired the rights from Neuron. I was not involved with these trials. I was involved with a smaller trial run at Boston Children's Hospital. And this was a major trial for a rare disorder, 184 girls across 10 sites. And it showed a significant improvement on girls aged 5 to 20 for 12 weeks. So many decisions have to be made for a clinical trial. How much drug to give? How do you go from a mouse to a human? How long? But it worked. And a year and a half ago, this drug, trofinetide, was approved by the FDA as the first treatment for Rett syndrome or for any neurodevelopmental disorder. So that was a big success. But how much time do I have? 5 minutes? Okay, because scientists cannot just stop there. What next? And so what next is mutation-specific therapeutics, because synaptic development starts early. MECP2 has pleiotropic functions. It not only functions in the postnatal lifespan on synapses, as I've been telling you, but synapses start prenatally in the human brain. And MECP2 has already come up in the human brain by the time a child is born. How might we study those and the effects of different mutations? We already suspect they are not identical. They may have some similarities, but not identical. So then we know that mice are not very good models for early human development. Again, you are lucky to have Dhananjay here. He'll tell you in great detail how the human cerebral cortex has a ventricular zone where there are proliferating units, but then an inner and an outer ventricular zone and a much more elaborately designed cerebral cortex. So we need to use organoids. And again, you have Pankaj here who will tell you a lot more about organoids, but for the last 5 or so years, we have been doing a lot of work with organoids as models of very early human development and the impact of early MECP2 mutations, particularly derived from patients or induced with CRISPR with isogenic controls. So what do we find? Very quickly, Tatsuya, very talented postdoc in the lab, has made organoids with two different mutations, a mutation in the NCOR interaction domain and a mutation just outside of it. And this is a missense mutation, and this is a truncating mutation. And they have very different effects. We know that because we can label cells in these organoids, we can label synapses, we can label networks, and we can image the activity because our goal is to measure synapses. And how does MECP2 affect connections and networks? So we can do that using multiphoton imaging and imaging labeled cells in human brain organoids labeled with GCAM, which is a calcium sensor. So organoids are remarkably mature at 3 months of age. They are in effect equivalent to human fetuses at 3 months of age. And you can see the richness of connections. We can image functional responses. We can characterize functional responses in these two mutation types, including their isogenic controls. We can characterize these responses. We can show that the mutant organoids in both mutations have reduced frequency of spikes, reduced correlated activity, and they have synapses that have glutamate and GABA receptors because blocking these reduces the spiking activity. We can build networks of the connectivity between neurons using ideas from graph theory that is also used for the analysis of fMRI and EEG data in human beings. And I'm not going to go into all the details except to say in my last couple of slides is that there are multiple properties of networks. And small world networks represent a balance of local and long-range connectivity that can optimize function or describes human function relatively well. But in the organoids, you can see that while the wild-type isogenic organoids do show small world connectivity, meaning a balance of local and long-range connections, the mutant organoids have different local connectivity. This mutation, the missense mutation, shows much reduced connectivity in the local scale. The connectivity is far more long range and random, and small world connectivity is the correlations divided by the path length. So it decreases when path length is high, whereas in this mutation the small world connectivity has gone up, meaning that is much more focal connectivity. So there are different types of changes due to the mutations at the network level. What does this mean for therapeutics? So while there is broad similarity, these neurons are not firing too good, there are different kinds of changes in the connections. We can do RNA-seq. We can ask, what are the molecular phenotypes of one mutation versus the other? In one mutation, there is upregulated HDAC. In the other mutation, there is downregulated GABA receptors. We can ask, are these potential therapeutics? Yes, you can use HDAC inhibitors and ask, does that restore connectivity in the R306C mutation? And it does. The small world propensity goes up when you rear organoids with this HDAC inhibitor. And you can ask, do GABA agonists counter the changes in this mutation? And it does, that the higher small world propensity comes down in the V247X. So this gives you a glimpse of the future, that based on the mutation type, we believe we can propose therapeutics based on the patient-derived organoids. So in conclusion, loss of MECP2 affects function and plasticity of excitatory synapses via downregulation of synaptic signaling molecules in Rett syndrome. IGF-1 restores function, and the tripeptide form of IGF-1 is the first FDA-approved mechanism-based therapeutic for Rett or for any neurodevelopmental disorder. Then mutation-specific analysis shows that neuronal connectivity, functional architecture are affected differently in at least 2 mutations that are actually pretty close to each other in space, but affect different subsets of the gene. And different molecules, an HDAC inhibitor and a GABA agonist, partially rescue connectivity. And looking even more far, I think that single gene disorders may well yield to gene therapy, but I'm not going to talk about that. And I'm going to close here, and thank you very much. I want to thank many people in the lab. That's my current lab. These are people who were in the lab previously, who contributed to the work I presented, and we have many collaborations.
Questions?
Sorry, I went over by 10 minutes. Yeah.
Really nice talk, Dr. Mriganka. I just wanted to ask, like, how long does it take to grow those organoids?
3 months. We have to grow them to enough time that not only do they proliferate the neurons, but they make synapses. Because our whole hypothesis is based on synaptic dysfunction as the principal target of Rett syndrome.
And
so it is difficult to see synapses too much earlier. There has to be 3 months is a pretty long time for organoids, though many people have grown them for many more months. It's just that the inside of the organoid, I'm not confident is viable because the growth factors and the molecules that you are growing them in need to get to the inside of the organoid. So the inside of the organoid begins to die. So 3 to 4 months is what most of our work is done with.
Okay, and my next question will be, then can we make it like patient specific, like for
100%, 100%. That's the idea. These mutations are patient specific mutations. In fact, in both of them, we obtained the cell lines. We obtained, skin punches from a company called Cortical. Ask your colleague. He will tell you in great detail. And then we CRISPR'd out the mutation to make isogenic controls, because everything else has to be the same except the mutation. And that's your control. The control cannot be some other person. So you have to make sure that that's what I mean by isogenic control.
My name is Dhruva, very nice talk. So in the Rett syndrome, what is basically the cause of the microcephaly? Is it due to the reduction in a cell number, or is it reduction in a cell size?
Very good question. It is a little bit of both. It's a little bit of both. And we have published on how neural progenitors are trapped in the progenitor stage and there are not enough neurons made. But secondly, neurons are smaller and the dendrites are smaller. So 2 reasons.
What about when you treat with IGF-1? Does it restore the cell size or?
No. We are treating with IGF-1 in a mouse that is already 2-3 weeks old. So early development has gone by. What is left to happen, that's why I started with my slide. What is left to happen in a human 2 to 15 years old, early development has gone by. No more neurons are being born. All the neurons we will ever have, we are born with, except in 2 regions, olfactory bulb and dentate gyrus, a little bit. But from that point on, it's all downhill. At my age, it's very downhill. I'm losing neurons all the time, man. In this talk, I must have lost 20 neurons, if I calculate right. So that's not good news. But in response to your question, new neurons are not being born anymore. and the architecture of connections is pretty much not changing too much. What is changing is local synapses. That is the point of our hypothesis. If we are ever going to have a therapeutic, it's got to target synapses. Because long-range connections and the numbers of neurons, dendritic architecture even is not going to change so easily in a brain that is fairly mature. It's not very mature, of course. It's still a child, but that's the idea. How should you think about therapeutics?
Thank you.
You can only think about what is to come, not what has happened. And time's arrow goes in one way. You've got to intervene with whatever is left to do.
Very nice talk. Is there a reason to think that MECP2 and the changes associated with the synapse will only be limited to the central nervous system and not in the peripheral nervous system?
Oh, not at all. I think that peripheral nervous system can have a big impact. The interesting thing is that the understanding of what does MECP2 do in the periphery seems to be not very much, though that could be wrong, because mice have been made with MECP2 knocked out everywhere else except in the brain, and they seem normal, and only in the brain but not everywhere else, and they are devastated. So a full MECP2 knockout is very equivalent to just a brain MECP2 knockout, including the systemic phenotypes such as heart rate and respiration and movement. You could attribute those to muscle atrophy or to peripheral reasons. Not so. These are central reasons.
Hi, that was a great talk. So IGF-1, I mean, is it receptor dependent perhaps, right? The mechanism of action.
IGF-1 activates the IGF-1 receptor, which is like a tyrosine kinase B receptor, so, like for BDNF, and it activates specific signaling cascades.
So that's the only ligand? I mean, that's the only
Very good
ligand.
question. IGF-1 has greater binding for the IGF binding protein than for its own receptor. That's why small changes in the binding protein lead to big changes in IGF-1 availability, which is why we believe IGF peptide works. It doesn't activate the receptor. It's a whole different molecular architecture, but it does bind to the IGF binding protein, thereby freeing up IGF-1. IGF-1 receptor can also be activated by insulin. And there is the insulin receptor, and there is some promiscuity. These are not very high affinity receptors. That's my point.
So then that pathway can also...
It is possible. It is possible. Yeah. My life is finite, so I can only study so many things. Yeah, but it is true. There are possibly many pathways that could activate IGF-1, and frankly, I'll never claim this is the only way to repair Rett syndrome. On the contrary, I already gave you 2 examples of HDAC inhibition and GABA that are other pathways that all play into circuits.
Thank you.
It was a great work. So my question is related to the ARC protein. So is that the synaptic pruning happens a lot till the adolescent stage, and then there are a little bit permanent connections that are being formed. So the ARC protein, is it also temporarily regulated during these developmental stages that is controlling the synaptic density in the developing brain?
Yes, ARC is a very important protein that is expressed during development, and there are papers that you can reactivate the critical period by upregulating ARC. ARC actually comes down during development a little bit. So the shaping of synapses is very prominent during critical periods of development, or at least during early development. In the visual cortex, there is a very prominent critical period. In the human language areas, there is a very prominent critical period. We know this from looking at our children. Language has to happen at a certain point in life for it to be prominently displayed. Otherwise, it gets a little bit more complicated, right? So there are critical periods for many things. And one of the drivers of the critical period, there are many molecules. ARC is one of them. Upregulated ARC followed by downregulated ARC, or growth factors like IGF-1 or BDNF, they get slowly upregulated to close off the critical period.
I mean,
So these are complementary ideas, and I kind of went over those quickly.
so there is a possibility that if we can reactivate the ARC protein in some areas, you could actually improve the.
You could activate plasticity. In fact, that has been shown. You can overexpress ARC in the visual cortex, and you can reactivate the effect of eye closure as an example of reactivated plasticity. Yes,
Thank you.
absolutely. Yeah.
It was a really nice talk. I wanted to ask something related to ARC protein only. So what happens in a knockout or a knockdown mouse where the ARC is not present? So would it lead to hyperactivity in neurons, something which might be also seen in a sleep deprived mouse?
Yes. If you knock down ARC, you have removed a very important mechanism for down-regulating AMPA receptors at synapses. It is not the only mechanism. There are other things that happen. So ARC knockdown mice live. I forget whether they live for the full length of life, or I think you can do conditional knockdowns in the experiments that we did. This was many years ago in collaboration with Suzumu Tonegawa's lab, which made the mice. But and that's what I showed in my ARC experiments. So the ARC knockout mice do live, but their plasticity is severely affected. You can survive without having your brain change as much as a wild type mouse.
And also, are they hyperactive in terms of their behavior or the activity?
You know, I don't know the answer to that. Yeah. I don't have too much ARC going on in me anymore, so I don't remember some of these things. But it's in our paper. There's a paper in Cell from 2008 in which we did these experiments that I described.
So that was a nice talk. I had a question with regard to the IGF-1 peptide treatment. So I was wondering if there are any selective retention and phenotypes following the treatment. As you pointed out that the number of neurons you can no longer regain, but just the connections are affected by.
So are you asking whether there was a full rescue or only a partial rescue?
And if
the answer is it's a partial rescue. And because the animal is not normal, it just lives longer, moves more, and breathes better. These are just phenotypes that our paper has described, but it is not a normal mouse because development is a one-way arrow of life. And we are intervening at some point after some development, or a lot of it has already happened, and it's difficult to rescue those. Particularly the structural changes are notoriously hard to rescue. Functional changes can be rescued. Why? Because the structural substrates are more amenable to change, which is the synapse. And the synapses have genes that are activity dependent, and they regulate the influence of activity on changing synaptic structure and function, and you can intervene.
Right. So my question was more specifically towards the human trials where the peptide is being given, and whether there is retention of specific phenotypes in those humans following treatment too.
Oh yeah. This was the human behavior and phenotypes had to be rigorously described, and I went over it very quickly. There are two tests that were done for this clinical trial. One is called the Rett Syndrome Behavioral Questionnaire, or RSBQ, and the other is the Clinical Global Impression Scale. I don't want to offend our child neurology colleague here, but they are amazingly simple, where a caregiver fills out a form. Is this child eating better, sleeping better, this, that, and the other, on some scale? And the clinical global impression scale is a clinician looks at a child or talks to the caregiver, and within 2 or 5 minutes has to fill out the form. But it's something, because these are very sick kids. You put them in a scanner. You can't even do EEG with them sometimes. But based on these ideas, based on these things, and for whatever it is worth, is done blind. The clinician does not know whether this child is getting the drug or not. That's a gold standard. Sometimes the real gold standard is a double-blind placebo control, meaning one group gets the drug, another group gets the placebo. So that's blind, and the clinician doesn't know till the code is revealed. But then double-blind means that the patient or the clinician doesn't know, and placebo control means that these are the two arms, and then you switch them. So the people who are getting the placebo now get the drug, and then you go, and then you break the code. So these are different ways of doing clinical trials. But so even humble methods of assessing, particularly for rare diseases like this, have validity if the trial is designed correctly.
Thank you.
Thank you. I think I'm the only odd one out here, being on the clinician side and handling these patients for last 3 decades. So I think it was an amazing talk. And that trial which we were done was actually with Neuron Pharmaceutical Cerezotan. It was not the IGF, and that's why it was a negative trial and never published. It's very difficult to do trials with these patients. Just wanted to ask you something that not with red, we're completely looking at it, and obviously with the other pathways coming up, so we can repurpose so many drugs, because generating new drugs from the new molecules is difficult, rather than you can repurpose the drugs. In the autism part, like autism, how do you think, as being your 5 decades of neuroscience, how much role does epigenetics play? Because I personally feel as a from the clinician side, that once we understand more of epigenetics, that is going to be a game changer, and we may be able to get some therapeutic intervention targets, we have that air pollution exposure, pesticide exposure, all what is happening, which are actually adding on. And even the simple thing like stress, because we've also studied on sperm epigenome for fathers, and we have found them having methylation defects. So all those things are going on, which are adding, and that autism is now becoming into a global epidemic. So what do you feel about the epigenetics?
Okay. So I'll give you my sense as a non-geneticist. Epigenetics and environment are two different things. The environment can exacerbate or offset genetic risk factors. Okay. The thing about rare disorders, as you know better than me, is that only one gene, but is highly penetrant. That if I have the MECP2 or the Fragile X mutation, chances of me having that disorder is very high, regardless of the environment. But the more complex the genetics, the more the role of environments, and indeed air quality or stress and many other things are known to play a role, not only for developmental disorders, but certainly for degenerative disorders later in life. So the risk factors for Alzheimer's are actually ApoE4 or presenilins, but the number of people who have Alzheimer's is very much more than the number of people who have the risk factors, and that's because life has happened. Now, epigenetics. MECP2 is an epigenetic modulator. It binds to methylated DNA, which is a mechanism for transcriptional regulation. So genes that are causally related to neurodevelopmental disorders can be epigenetic modifiers.
Fascinating, Mriganka, as always. So, one question that I was wondering, so how do you see, are you there yet in terms of using the organoids and development of these network measures in deciding where to intervene and where not to intervene, maybe allow more of not drugs, maybe something other kind of therapies, behavioral therapies, maybe a good solution. Are we there yet?
Yeah. In theory, but you know, I have a hammer, so everything looks like a nail to me. And my hammer is, we can grow neurons, we can measure them, we can analyze them, and we can manipulate them by throwing substances on them, or by genetically, CRISPRing one thing or another. Okay, so those are the tools that I have. And our goal is to discover molecular therapeutics for disorders. You are asking, can there be other therapeutic, behavioral therapy, let's say. In fact, in Rett model mice, if you just rear them in an enriched environment, the way we rear our mice are horrible. But if you just put them in a cage that is 3x by 3x and you put some toys in it, the mice get better. Now, the open question to me is whether this is applicable for humans. Human beings, to the extent possible, are not reared in impoverished environments, though poverty is a very big risk factor for brain disorders. Our socioeconomic status or nourishment, exactly. So perhaps, but you don't have to have a brain disorder for me to tell you this. All of society should be properly nourished and properly housed. But would that reduce the incidence of brain disorders? I think so. So this also goes to the issue of environmental factors. But I think training, targeted training, to be more focused about your question. Can it help? I believe so. Because what does training do after all? Training reinforces synapses. And synapses are the absolute ultimate target of therapeutics for almost all such conditions. Yes, please. I'm sorry to keep you, but I'm happy to be here for as long as you like. This is what I do. Yeah.
Thank you so much. Very interesting talk. I have a follow-up question with those conversations about prenatal nutrition. How does that impact any of this condition? Can it be prevented? I mean, if the nutrition can be improved in mothers.
Definitely. I think that malnutrition is a crime against humanity because it is a treatable, problem. Even in the United States, one of the richest countries in the world, there is significant malnutrition. There was a study that 30% of children, particularly children in divorced families who live with, impoverished mothers, have not because of the lack of resources, lack of money. Okay, so this has been actually proposed as one of the reasons why the incidence of autism has been rising in the United States. So in these ways, brain disorders are as much a social problem as a molecular genetic problem, okay? Because I fully agree that the molecular genetics follow the environmental contribution to perhaps risk factors that exist. But there are risk factors in all of us, So to that extent, no question that nutrition and socioeconomic status is a huge cofactor. Many studies have been done. The WHO, and I was part of a WHO study in terms of disability affected life years, dailies, which is what they measure. And poor countries are heavily impacted. It's not because the genetics are bad, it's because the socioeconomic conditions are bad.
Nice talk. I have one question. So the intervention was done over different age groups of girls. Is there a data?
Because that's who showed up.
Yeah,
not because you are selected. Once you start a clinical trial, you can't just say, I'll just take 2-year-olds already it's a rare disorder, and then you have only 2 people left.
Yeah.
So you have to broaden the criteria.
So was it that the people with the girls with a younger age, you got better? So just instead of just making their life better, was there was a developmental aspect to that as well?
Yes, very good question. Did younger girls who got the treatment get more better than older girls who got the treatment? I think there is a little bit of signal on that, if I remember right. But the paper has been published, and you should read it. It's in Nature Medicine, and the first author is a colleague called Jeffrey Newell, N-E-U-L. Yes.
Mutations in for the Rett syndrome, they are germline?
Yes.
So theoretically, in pregnant mothers,
you could do testing.
You can do testing.
You can.
So has anybody?
In fact, there has been talk about this, but it's an ethical minefield. Where do you stop, and how much do you test? So for certain things like trisomy 21, it is accepted.
So in mice, have you tried to give the peptide
in utero?
Pregnant in utero?
Yeah, we haven't. We haven't. And it's an interesting question. As a scientific question, we have thought about it, but we haven't. But maybe we should. So, but it's a very good question. We haven't.
Okay. And have you tried? I mean, this peptide probably is given as nanoparticle, right?
It is given as an emulsion. It is a nanoparticle, but it is as an emulsion, and it is given by mouth twice a day.
Yeah. The problem with emulsions is that there is also a decay, and it goes
100%.
So have people tried to do it with a hydrogel, with a slow releasing peptide?
This is a very good question. Hydrogel, or to reach the brain, nasal spray, etc. And the issue is, you know, I'm not an expert in this, but there have been conversations. And I have not been involved in the delivery at all. When this company approached us, they already had this emulsion going, and they wanted to do it. So we never even insisted on a patent or anything. We just gave it away. We just gave it away. I get nothing out of any of this. So, but it's a rare, devastating disorder, you know. So you need a company or somebody who's doing the trial to try different things. And maybe there's a meeting actually in 2 months where the company has invited us and many others, and maybe this will be discussed.
And last, can I have one more?
Yeah, sure.
So since you are now moving to the next generation of therapy using patients, and I understand these are with chorial cells where you are actually making the isogenic
But we could
lines.
actually also have access to patients themselves directly.
Yeah, why not take the blood? Why not take 5 ml of the blood and do an iPSC and give it back to the patients in 4 months' time? A corrected
You mean
one.
give back what?
A corrected version of...
No. It doesn't work like that. You mean take the blood, correct it, and put it back? No. That my understanding is that it doesn't, you know, there is a blood-brain barrier, and stuff in the blood doesn't cross over into the brain. The brain has gone to great trouble to protect itself from external influences, and that works against us.
So how do you think that your work on the next generation
Gene therapy?
Gene therapy.
Oh yes, very good question. So the peptide, for instance, how does it get into the brain? It's an interesting question. There are some transporters for molecules across the blood-brain barrier. And one of them is a transporter for insulin and insulin-like growth factor. That was the long answer to what is the biology of the receptor system. And so sugars, irons, growth factors are transported by special transporters across astrocyte end feet, which form the blood-brain barrier. Most molecules don't cross. So that was another reason for thinking. I didn't want to get into so many details, but for thinking about IGF-1 as a particular molecule that could be delivered through the muscle, like we did in the mice, or through the bloodstream, as the humans get, because this is absorbed into the bloodstream. And it gets into the brain through this by hijacking existing transporters. Now, any drug that we use for future therapy will have to be thought of in this way. Gene therapy has to be delivered into the brain. If you deliver gene therapy into the bloodstream, if you deliver, let's say, an AAV vector containing some correct gene, it'll never get into the brain. So in fact, the gene therapy has progressed. I'm not a part of it. Unfortunately, and as I was just saying to her, a patient died from the AAV, which was supposed to stay in the brain, and the brain doesn't have receptors for AAV9, but the liver does, and there was a flare-up. So it did cross out of the brain in ways we don't understand. Yeah. So gene therapy is not a magic, but it will likely work with enough people in your lifetime, if not mine, for sure. But there is a special challenge with MECP2, which is that there is not only an MECP2 deletion syndrome, which is Rett syndrome, but there is an MECP2 duplication syndrome. Too much is bad too. So you need to titrate the level very accurately. And that's not a trivial issue for gene therapy. Unlike for SMA, for spinal muscular atrophy, for which the first gene therapy drug has already been approved. But you know, I regret having not had some control over this molecule. You know what has happened? And this is the tragedy of healthcare systems for profit. This company makes this drug, and do you know how much it charges for 1 year of supply? $600,000. He's almost right. Yeah. You take it 3 times a year for 2 months at a time, 2 months on, 2 months off. And insurance covers it for people with Rett syndrome. But there is so much hunger that other children, so this company is also running the same drug without any evidence whatsoever for Fragile X or for Angelman syndrome. Why should they do this? You would think rationally there should be some biology and show some evidence that it will work and why and this and that. And I showed you the logic. But if you have a child with Fragile X, you are so desperate, you mortgage your house to get $1 million to buy this drug. And people are doing that. This is a crazy game. This is a crazy game. If I could, I can tell them I bought a gallon of IGF peptide from Peprotech, you know, for $100. They should just get it and try it if they want to.
But I thought the regulation in US must be very strong, and I thought this is happening in India only. I didn't know that.
India models itself on the United States. What are you saying?
So if there's no further question, let's thank Professor Shur again one more time. Before we conclude, I would like to invite Professor Sagar Sengupta to felicitate Professor Shur regarding this Ramamurthi Memorial Lecture. Okay.